Hello everyone. So today I'm going to be guiding you through the analysis of a dot plot within our Phoretix array analysis software. Now, Phoretix array is designed to analyze any image from an experiment based in an array format. So again, what I mean by that is kind of a square with wells in it. So it could be a 96 well plate, it could be a DNA microarray. In this case, it's a dot plot where you've got this array of different intensity dots, and the intensity of those dots means something. We want to convert that into data because the response, the intensity, is something that if we measure it and quantify it, it could be useful to us. So a dot plot is kind of the much quicker, easier version of a Western blot, whereby you're not super interested in sensitivity or identification as much as you're interested in kind of presence or absence. Is the protein or the DNA that I'm interested in present in this sample or not? So whilst Phoretix 1D is obviously concerned with quantification and analysis of immunoblots like northern, southern, eastern, western blot, bands, lateral flow tests, things like that, array is more focused around quantifying circles and comparing them to each other, but there's a lot of overlay in terms of what people want to do with their experiments once they've got the image. So a lot of the rules from Phoretix 1D apply here. If you've not seen the videos on Phoretix 1D, if you only do array-based experiments, then I'll quickly go over them. This screen here is where we import our images from our scanner. So in this instance, for this demo, I've got a membrane from a dot plot. So this has been scanned on a gel doc device or a typical scanner and produce this image. Now we support all industry format images, so TIFF, bitmap, PNG, JPEG, all of that. We support pretty much any image type or any industry standard image type, but we would always recommend that people use a .tiff image, simply because they are uncompressed. We don't have to deal with compression artifacts. It's as raw data as we can get. So I've imported the image here and I can see that the data is looking good, but it's quite hard for me to work out where I've got responses or not to my sample within my wells, simply because the contrast needs a bit of an adjustment. So this image view is where you would bring images in. It's where you can edit images if you want to. So you can crop, you can rotate, you can do inversions, things like that. All of that is automatically built in the software for you. So you don't, it's an all-in-one package basically. So what I'm going to do here is I'm just going to set the contrast range to full. Now it's really important to understand here that all of TotalLab software takes measurements for volume, background, wherever it may be, directly from the image, straight from the raw file. It does not take it from the view that we have on display here. So this ability to change contrast, change gamma, change how this image appears to us, the user, is purely for the user's benefit to be able to do things like define where wells are and grids are and things like that. So even if I took a measurement when it looked like this, changed it like this, took the same measurement, I would get the same results. It's really important to know that whatever you do here doesn't change your results, doesn't change the numbers, just changes how you can visualize the data on your computer, with your eyes, in your lab. That's what that's for. So it's also worth noting at this point that you can expand all of your 3D view. You can copy this image to your clipboard, so you can put it in a report, you can put it in a poster, a presentation, you can do whatever you want with this image. And you can also export them for presentation, for kind of papers and things like that, directly within the software. Again, you can perform operations like crop. You can add annotations, but I've not added any yet. You can put on distance scales and intensity scales for your measurements. You can do all of that from straight within the software. So you don't need any other software to edit your image or kind of export it in any way. So the first step in our analysis workflow is defining where we want to take our measurements from. So we do this by defining a grid. Now, older versions of our Phoretix array software, because it's been around for a long time, used to feature automatic grid detection, and we moved away from that because it simply wasn't as reliable as giving users really good tools to generate their own grids. it wasn't coping very well with non-standard, non-square grids. So we thought the most bulletproof way of this being able to work for everyone is by giving people these little drop downs and allowing them to select the size of their grid. So I've got a 7x4 here, and I'm just going to left click and drag this along. And you can see that I've dragged it far too large. Oh, that's a 4x4, never mind. So we've got 4 rows and 7 columns. Left click in the center here and drag across to fill out my grid. And you can start to see my measurements appear at the bottom as well. Now, if you are using an industry standard 96 well plate or 384 well plate, we've got these handy buttons here that you can just click and add that on. Now you'll see here, Phoretix Array does support multiple grids, and you can compare your results between grids. So if it fits within the context of your experiment, if you've got multiple grids within an image, and some of these high resolution microarray images can have, lots of different grids from lots of different samples, from a kind of a high level overview, we support that as well. Multiple grids, multiple different sizes. The grids can all be different to each other, but we can take measurements and compare measurements from all the different grids. So although I'm using a simple dot plot within this demo, that's by no means the limitation of the software. It's just that it's quite an easy jumping in point to be able to visualize a dot plot, and then the tools that we've got to analyze that. So once we've defined our grid, let's say we're using a dot plot, but our lines aren't completely straight, which is kind of the case here. You can see that these dots aren't perfectly in line with each other. But what we can do is we can bend and adjust our grid to make sure that we're still taking measurements accurately from every well within the grid. So by clicking on Edit Grid, we've given the user the ability to edit the grid, essentially, to be able to stretch it around your results. Edit Wells allows you to actually move individual wells within the context of the grid. So even when you've edited your grid, if you really have something non-standard, you can move everything individually and make sure you're getting accurate results, accurate measurements. We can import and export your grid as well. If you've got a very standardized experiment that you do all the time, you always use 7x4, you can save that as a template and import it every time. So you don't have to go through this and create your grid every time. You can simply have that saved and import it every single time. One thing to note here is my circles, my measurement circles as part of my grid are quite far away from my data. So some people get very caught up on wanting to make, I've got to make my measurement circle as tight to my data as possible. And actually, it's much better to err on the side of caution and have a bigger area around your data, your kind of your sample, your signal, because we're going to use background subtraction to remove the value of this, the pixel values around here anyway. So what's going to happen is when we define our background subtraction, let's say we've got the area outside here and inside here, the values for these pixels are going to be so similar to that outside where we define our background, the actual measurements are only going to start really about here. So although our measurement circle is much larger than our data, we're still just going to be measuring at the pixel level when the signal comes in. And if we start to do that from here, we actually might miss a lot of our signal that is sitting on the outside of our well here. So if you are someone that's used Array before, currently using it, trying it out, and you're watching this video as a bit of a demo to try and learn its features, that's one piece of advice I would give you is always go bigger than you think you need to, because the background subtraction is going to sort that out for you anyway. Don't go super tight to your data, because you're probably going to cut off some of your own data by doing that. And that might be the difference between a positive and a negative result. We do also support square wells, so we can just turn this into a square. I don't quite know what that's for, but I'm sure it must have a reason to exist within the software. So someone must be doing something with square wells out there somewhere, and have asked us about it in the past. So the next step in the analysis workflow is the background subtraction. Now, we feature multiple methods of background subtraction. It's up to the user which one they prefer, which one they think is most appropriate for their experiment. So we have surface minimum, So this treats every well. Most of our background subtraction techniques treat each well as a separate well, and they attributes them a separate background. So surface minimum will scan the edge of your measurement circle here, so the pixels on the surface of your measurement circle, and it will pick the lowest pixel value in that circle and assign that as a constant background value. But it will do it individually for every single well. It means that your results are much more accurate. It doesn't assume that the background across your whole grid is completely even to a singular point, to a singular value. And then our edge average background removal technique again scans the outside of your measurement circle, but this time it averages all of these pixel values to give you an average background from around your well, and then subtracts those pixel values from all of the values inside of the well. Negative control is for if you've got a negative control or multiple negative control wells in your experiment. What you would do is you would click on them, left click, and then set your negative control as your negative control. And what it would do is it will take the volume from this well as background and remove it from every other sample. So if you do have a negative control in your experiment that produces some signal, you can remove that signal from every other well so that you're only analyzing, experimental results. Increases, changes that are down to your experimental protocols, your treatment groups, your different groups, whatever it may be, not just influenced by the inherent autofluorescence you've got in your system, for example, something like that. But we can also support setting multiple wells as negative controls. So what happens here is I left click and drag, and I can select these three, for example. When I set these as a negative control and I get them listed here, we take the average volume of these wells and set it as the negative control for your experiment. So if you've got a negative control in triplicate, of course we can support that as well within the software. We have the classic image rectangle background subtraction. So what we can do here is we can define, just as in Phoretix 1D, we can define a rectangle, and what the software does is it averages all of the pixel intensity from all the pixels within this rectangle and sets that as a constant background removal value. Constant background removal values, and I've talked about this in Phoretix 1D, are risky because they assume that across your image, top to bottom, left to right, your background is all the same, it's completely universal. So you can just take the same value from everything, from every XY position in the image, it will be the same. And that's very rarely the case in an experiment. It's very unusual for you to get completely to the pixel value level the same background across all of your experiment. So using adaptive techniques like surface minimum and edge average, and edge average is always our recommended background technique if you don't have any preference, if you don't know what to pick edge average. They're always recommended over constant value techniques like image rectangle. And of course you can select no background removal techniques, so you can just get the raw volume data that way. Although if you want your raw volume data, we do just show that within our columns anyway. You can just turn that on at the value that you want to see. So in terms of controlling which data you're looking at, what data you can see, we have the ability to turn that on or off within our grid view here. Oh no. So if I turn on my all wells view, if I click on columns, I can turn on or off different measurements that I want to view at any one time. Now, it's really important that people investigate this column section because they might think that the software is unable to perform these measurements, and it's simply that we don't have them all turned on all the time. That's because we don't want to clutter up this area with data that we don't know that the user wants to pay attention to or cares about. But if we simply tick these on, we can then export these directly to a CSV. So we can export all of this directly to Excel, or we can copy it to your clipboard to allow you to paste it into Excel yourself, paste it into MATLAB, R, whatever, further processing piece of software, sorry, that you want to use, you can export that to and you can bring it in. Put it into a database, put it into a LIMS system, whatever it is that you're using. We fully support these kind of generic export methods. The next step in the workflow is the ability to normalize wells to other wells. And the same logic applies here, whether it be a single or multiple wells, in that if it's a single well, we just take the volume of that well. If you add multiple, it becomes an average, and we can actually choose which normalization function we want to use. So it's either average or it's total volume of those multiple wells. And we can set the scaling factor as well. But basically what we're doing here is when we set these, I'll take these three as an example. When we set these wells as our reference wells for normalization, we tell the software the average of this is 100%. Give me all the values of all of the other wells as a percentage of that, or relative to that. So we can see here that our volume percentage normalized, we can see some are 140%, this is 119%, for example. And it's very useful for showing kind of upregulation or results relative to a control or a positive control, negative control, whatever it may be. So it's just another way of visualizing data and relating data within your experiment. Again, it completely depends on the structure of your experiment, but the tool exists there for you to be able to perform that work, basically. The next step in the workflow is quantity calibration. So quantitative extrapolation of your images into known values or with known values. So you can start building up your concentration curve and work out your unknown concentrations or masses, whatever it may be, of your proteins or nucleic acids if it's a dot plot, or if it's another type of array assay, you can do that as well. So what I'm going to do is I'm just going to add this well to my concentration curve, and I'm going to say, right, this was my 10 nanogram sample, for example. This was my 5 nanogram sample, and this was my 1 nanogram standard. And you can see from here we've got our calibration curve, and we can change which curve fitting type we use with this based on what we use within our experiment. We can export this, we can copy it to a clipboard, we can do pretty much everything with it you would expect from a powerful piece of software like Phoretix Array. And then we can see all of our unknown values relative to our standards, relative to our concentration curve in our data here at the bottom. Again, this can be exported to Excel, or we do support the ability to export PDF report from Phoretix Array as well, of course. The last step of our workflow is defining wells as present or absent. So this is basically a simple threshold filter to say whether we're considering it's a positive result or a negative result. So let's say I want to consider anything with, any wells that have an intensity higher than this well, I'm going to say worked for a positive result, for a positive response to a drug or something, whatever it may be. If I just select this well and click on threshold from selected, everything above this well's volume will be listed as green. And I can actually turn on a presence and absence table here that again I can export, and then I can do multiple as well, so I can average the volume of these two, or I can change it from here, or I can set it completely manually based on the normalized volume of these wells. You can see they go red if they're below the value and green if they're above. So red is absent, green is present. The final thing that I want to show you within this software is something that is prevalent within all of the Phoretix suite of software products, is the ability to save everything I've just done here, all of the analysis I've done here, as a protocol, which I can then share with colleagues, move between different sites, send an email. So what I could do is I can save a protocol here. That will give me a file, and then the next time I want to do an experiment that has a similar layout to this, so if I'm bashing these out every week or every day, if I'm doing hundreds of these kind of dot blots or array-based experiments, I can simply apply the same protocol over and over and over again, and it will do the analysis the exact same way every single time. So we can massively reduce the subjectivity and the interoperator variability inherent within image techniques by allowing the inclusion of protocols, which can then be stored centrally, have SOPs written around them. There is a 21 CFR and GMP compliant version of the Phoretix array software available. So yeah, you can share those protocols with friends. You can make sure that you're doing the same experiment the same way every single time. And I don't think I've shown you the annotations feature as well. I lied before. This is the last thing that I want to show you. Is I'm just holding control and clicking and dragging here. So I can add different annotations to label my wells, say things like, mistake here. So if there are any areas of, for a dot blot with the membrane-based technique, if there was, a giant cut here or a giant black mark or something, you could say, oops, I dropped it here, disregard these wells, that kind of thing. So you can include all this information. You can export it as part of your presentation images. You can export it as part of your PDF report. So if you want to keep a log of, any mistakes, anything that was out of the ordinary, by using annotations, you can include that, keep that in your experiment forever. So the next time you open it, you can see that immediately. And you can turn your annotations on or off if you don't want to see them within your exports, or if you want to keep them just for internal use. But they're there, they exist. They're a useful tool in some hands, in some circumstances. So I think that's all from me. As with all of our software, if you're interested in getting a free trial of the software to use in your lab with your own images, please check out the links in the description below. If you've got any questions, again, drop them on the video, get in touch with us here at TotalLab. We'd be happy to answer them. And until next time.